Okay, well, good afternoon, everyone. This is the final session of the spring series on water quantum biomolecule. Betony is going to bring together the presentations. All our speakers from the series are here today, so we welcome them, and we're looking forward to a very robust discussion after the presentations. And then Betony will introduce Wayne, who's going to give his personal reflections on what he's heard in this series, and so we're very much looking forward to that. So I think without further ado, Betony, should we hand over to yourself?
Thanks, Geoffrey. It's my pleasure and challenge to try and summarize the excellent presentations in this series. And thank you to all the speakers for your summaries, as I've used details from those. As always, there was a large amount of information to compress. So if I've missed or misrepresented anything, please let us know in the discussion. So the autumn series focused, as Geoffrey said, on water and its role in biological systems. And it's well accepted that water is integral to sustain living organisms. But what is less well appreciated is the novel behavior of water at its interface with various biological materials. And through the series, we explored the physics of water in different biological contexts and to what extent quantum mechanics might be implicated. Dr. Philip Kurian kicked things off with an introduction to the physics of water and the possible role played by water in the origins, organization, and development of living systems. He began with 5 features of water that make it, as he put it, wondrously weird and interesting in the context of life. These include its extraordinarily high melting and boiling points, the highest latent heat of vaporization of any known solvent, the fact that ice, which is the solid form of water, floats rather than being denser than its liquid form, and the fact that water has 3 phases across Earth's temperature. And finally, water's higher relative permittivity, or dielectric constant, than almost all pure liquids. He also discussed the relative strengths of various types of chemical bonds and highlighted the significance of different hydrogen bond networks. Philip then turned to origins of life research to illustrate the role that water may have played in this. He reviewed origins of life experiments and outlined how single critical events that are highly improbable can become almost inevitable given sufficient numbers of trials over time. He also took us through how nucleic acid, protein, and lipid precursors emerge from cyanosulfidic reagents in a primordial soup under ultraviolet light, and how a similar prebiotic chemistry might work on planets different to Earth. He also discussed how water might form supercritical fluids, which allows for the tuning and optimization of parameters such as density, viscosity, and relative permittivity. And finally, he discussed how water participates in collective interactions, which he very poetically called a terahertz dance around peptides and whole proteins. The dynamical hydration shell around proteins can create an extended dipole, which then acts as a sort of antenna in solution, and water can then mediate collective interactions such as Froelich condensates. And Philip also reminded us that the electromagnetic spectrum of light is a collective signature rather than the signature of any one molecule. And he finished off with a broad picture of the chemistry and physics of photosynthesis, aerobic respiration, and the role of aromatic heterocycles from interstellar media. Our second session had Dr. Ali Hassanali build on Philip's introduction to the physics of water by looking at this physics in specific contexts, particularly at interfaces, both from the point of view of theory and experiment. And I really enjoyed Ali's lecture for how it so clearly illustrated the creative interplay between theoretical modeling and experimental verification. He began by outlining how we can investigate the properties of water, either in bulk at interfaces and under confinement. And this includes looking at its molecular dynamics and electronic structure. And he also mentioned how we can use unsupervised machine learning as a tool for analysis. He went on to discuss how interfaces change the dynamical, structural, and dielectric properties of water before going into detail about water interfaces and the interplay of charge, pH and ionization, catalysis and electric fields, and then what this might mean for more complex interfaces such as mitochondrial membranes. Ali then went on to discuss how he models interfaces, specifically how he models simple water interfaces such as soap bubbles, He first took us through an example of the simplest model where he showed that adding a surfactant to create this membrane changes the orientation of water molecules and results in charge density, large electric fields, and low dielectric environment. He then speculated on the mitochondrial case, where membranes will have spatial and chemical heterogeneity, likely leading to variable electric fields. He took us through how we might try to model this by adding mixed surfactants, and he also reminded us that water was important, but so are the other things in the solvent. Ali then looked at what happens to water when it is confined and the extreme confinement necessary to induce notable changes, and whether any of these conditions of confinement might be met in the context of mitochondria. He then moved on to discussing the interaction of light and water, particularly UV photochemistry, and went into detail about photoexcited ice and radical generation. And finally, he speculated on the possibility of related effects in biology, such as light-induced protein aggregation. In the third session, Dr. Nathan Babcock introduced the possibility of quantum effects in biomolecules. He first introduced the basic concepts of quantum mechanics before relating these to simple biomolecules, as well as outlining Einstein's study of the quantum mechanical nature of wet matter. He then outlined the quantum physics of proteins, with special mention of the electronic and photophysical properties of central amino acids, such as tryptophans and other aromatics. Nathan then introduced the many ways in which water and proteins interact. In particular, he looked at electron transfer and how Marcus theory had to grapple with the role of the solvent and the solvent reorganization and its relevance to electron transfer. Nathan took us through his own research into electron transfer between proteins and the water bridge that facilitates this transfer. And I really like the fact that it was called a molecular breakwater. He also looked at the role that water plays in proton tunneling, enzyme catalysis, van der Waals forces in DNA, and intrinsically disordered proteins, as well as ATP synthesis. Nathan also introduced the idea of collective interactions mediated by water. It was, as ever, difficult to choose just one sample of the excellent slides presented, but I thought this slide of Nathan's was particularly illustrative of just how difficult it is to pin down the exact role of water in biological systems, especially given this range of timescales over which water-mediated effects can occur, from picoseconds to microseconds. Our final session turned its focus specifically to light and water and what this may have meant at the origins of life. Dr. Robert Fosbury suggested that the sunlight reaching the Earth's surface is both qualitatively and quantitatively matched to the energy requirements for life on the planet, and that this coupling is possibly mediated by water and tuned by evolution. He outlined the properties of sunlight, as well as the absorption properties of living tissues, which lack strong absorbers in the near infrared, but scatter very efficiently, with maximum penetration around 850 nanometers. And he went on to explain that due to the properties of atmospheres in sunlight and cooler stars, the emission measured in photon rate per unit energy peaks at a photon energy of 0.75 electron volts. And this, surprisingly, is very close to the metabolic action potential of all life, which has a mean value of 0.66 electron volts. The absolute value of the photon flux would provide significant energy to catalyze electron transport chains throughout much of the biosphere. So in essence, he suggested that water could be an important chromophore to mediate the photon biomolecular interaction. Professor Alistair Nunn then concluded the series by taking us through origins of life research and how light and water may have played a role. He outlined how life is electrical due to its reliance on a flow of electrons leading to proton gradients. Life is also modulated by light while producing its own photons. He also explained how adaptive thermodynamics and quantum mechanics may have played a role in getting life started as a far from equilibrium, self-organizing, dissipating structure, and how this is reflected in life's apparent wastefulness due to uncoupling and futile cycling. Alistair went over the history of electromagnetic radiation in biology, outlining the concept of a morphogenetic field and Gerwich's discovery of mitogenetic rays emanating from onion roots, which laid the foundation for bioelectromagnetism and photobiology. And this history has also been turned towards trying to understand why red and infrared light seems to be medically beneficial. Alistair also took us through the other ways in which light interacts with biological systems, including the important role of melatonin. He then combined this thinking about light with thinking about water. Water is also an efficient absorber of far ultraviolet, UV, and red and infrared, while being transparent mostly to the visible spectrum, which may have been optimal for life to get started, given the proliferation of chromophores in living tissue. Water is the dominant metabolite in biochemistry, and its viscosity can be altered by red and infrared, as well as electric fields. Alistair then suggested that this all points towards alkaline thermal vent theories of life's origin, or something similar near to the sea surface. He expanded on the details of this, which I won't go into here, but if you missed the session, the video is now uploaded to the Guy Foundation YouTube. It is now my pleasure to hand over to Wayne Frasch, who is going to draw some threads together and generate questions for our discussion. Professor Wayne Frasch is the Biomedicine and Biotechnology faculty group leader in the School of Life Sciences and part of the Biodesign Institute's Center for Mechanisms of Evolution at Arizona State University. His research focuses on bioenergetics, the molecular processes of biological energy conversion, and he pioneered single molecule methods that provide a detailed understanding of ATP synthase molecular motors. He has over 80 peer-reviewed publications and 11 patents, as well as a number of awards, and that's just a sample of his biography. And it's my pleasure to hand over to Wayne.
Thank you, Betony. I have a greater appreciation for how difficult your job is to do this. So I've organized things a bit different, knowing how Betony goes through and makes a summary in order of the presentations. And I went through and reorganized things as to topics that were covered by multiple people, by multiple talks. And so, and of course, Geoffrey has mentioned a couple of times he wants my personal view of things. So I'll give some personal backgrounds of my experience of when I was in the room when it happened. So one of the things that three of the speakers spoke of was the effect of light and water. And so Philip mentioned how maybe if you're on another planet, one of these big gas giants where there's much higher pressure and temperature, where there's compression, you may have a whole different set of molecules that are used for energy conversion and the origin of life. Here on Earth, it's defined by a water environment that's separated by very, very thin membranes that have to maintain and hold up a huge amount of charge across there. So in photosynthesis, you see at the top, they're the electron transfer reactions, and they go from, you're taking electrons that are positive 1.2 volts, and you're moving them up across the membrane from P680 to P680 plus to, minus 0.8 0.8 volts, and then you need a second photo system in order to get them up to minus 1.2 volts. That then goes downhill in energy till you finally reduce NADP. And of course, this requires the light that was discussed by multiple people. And so this cross-section of what photons are able to do this, and how much the membrane can take as far as getting across the membrane. Ultimately, the electrons come in higher plants from water, where water gets oxidized to molecular oxygen. In so doing, it releases 4 protons to the one side of the membrane, and that's done by the oxygen evolving complex, which is a 4-manganese system. So then, of course, the oxidative phosphorylation just takes the analog NADH that's reduced and converts it back all the way, taking those electrons from a high energy back to the low energy side, where cytochrome oxidase will form water from oxygen. And early photosystems did not have the oxygen evolving complex. So they were not able to do this oxidative span of going from an extremely strong oxidant to an extremely strong reductant. Then again, Betony used the same slide that Philip had mentioned about how every protein that's water soluble anyway has a number of water molecules that surround it, and several of the speakers spoke about this and how those water molecules work. And I would say that one thing to note about this is that there is a process called salting in and salting out that's been known by biochemists for many years. And it takes, this is not pure water, but it has to have a small amount of magnesium chloride, sodium chloride, or potassium chloride that can also exchange on and off the surface of a protein to keep it folded and keep it working. And so this is also a very important part of this. And then, in fact, Philip did mention something about this, of how if you change the salt concentration, that you'll change the structure and cause proteins to come together. So when you're salting in, you're going from, distilled water to this small concentration, often like 5 millimolar of salt present there. And this keeps the proteins stable, where the charged and polar residues are mostly on a water-soluble protein facing outside, and this allows the hydrophobic groups to come together on the inside and hold the protein together. And as you change that, you'll find that you increase the salt to a point where you're salting in the protein concentrate. The protein will actually pull together. That's the salting in part. You keep going higher and higher salts, it'll start to salt out, and the salt in the solution will pull the water away from the surface of the protein, and ultimately it will fall out of solution, aggregate. This has been used for years now to get crystals of proteins for crystal structures. And so these protein hydration layers, as I said, they were discussed by several speakers. And yes, there is a layer that's bulk like water, that where the water molecules are floating freely, but then there's also these surface layers, biointerfacial water here. And I think this was Ali's slide. No, this is Nathan's slide. And so the other thing to note is that at the scale of a protein, the water, especially on these surface layers, is perceived by the protein to be very viscous, so that to drive a conformational change of the protein can actually be a difficult process that requires energy input. If I can go on to the next slide. But I'm sure I'm bringing these topics up because they covered by several people, and I think that they're very good points of discussion. Nathan talked about different computer models of liquid water and how this can affect the folding, and especially of disordered proteins, which I found really, really interesting. I had not heard this before. And yet, of course, it's all great when proteins fold well, but there's some proteins that need to be intrinsically disordered in order for them to work. There's the inhibitory protein of mitochondrial F1FO that is disordered until F1 starts hydrolyzing ATP, and then it folds into a helix and gets sucked in like a spanner in the works to stop the ATP hydrolysis-driven rotation. So this is also important for proteins related to Alzheimer's as well. And the fact that there are different kinds of water I found really interesting. I was really intrigued by Philip's discussion of this process, and I'd like to hear more. So were these pulses of light, were they pulses? Were they short pulses that were modeled to get these orientations of the water that then moved through space? And it I would just really like to hear more about that. And it seemed like a really cool piece of work that they did as far as molecular modeling. And again, I think this is Alistair, that that there was this paper that he saw where right near a membrane you'll have this exclusion water, whether it's myth or reality, but it actually forces certain proteins, other things away from the surface. And again, this seems to, I had not heard about this before, but it seems very interesting to learn about, and it could be a good point of discussion. Next slide. Nathan brought this up. He probably didn't realize that I did my dissertation on the oxygen evolving complex. I worked with George Chenet, who had only a couple years before discovered that the oxygen evolving complex contains 4 manganese, and ultimately George Chenet also determined that it has a calcium in there. My work in part led to the discovery of the chloride being part of this, and I found out, I continued to work on it after becoming a professor, and I found out that when you get rid of the chloride, if the chloride is removed, then you get a short circuit, and instead of going from water, from water to oxygen and 4 protons, that it actually short circuits and makes hydrogen peroxide that then goes and destroys photosystem 2. And so the chloride is very important in this. And I had not kept up with it since I started working on the ATP synthase. So to hear this discussion about these protons, so photosystem 2 processes 1 photon at a time. The photon comes in and moves 1 electron across the membrane at a time, but it takes to oxidize water to oxygen, it requires 4 electron process. So it's this manganese cluster that holds those oxidizing equivalents, and it actually causes the removal of 2 of the protons in the course of doing that. And it's just an amazing thing because it's every time an electron is removed, it's stable on the order for seconds, which is, it's an amazing enzyme. So it's what is going on right now as far as understanding what the involvement of the waters are. Marilyn Gunner and Gary Brudvig are really exploring this, and they have a couple papers just in the last couple of years about what these waters are doing as this enzyme works. It's responsible for almost all the oxygen in the atmosphere, actually. So Ollie talked about what happens when water is in confinement, and this was also brought up by other speakers as well. And it changes the dielectric constant and density oscillations. And I found that very interesting and overlapping with a lot of questions that I'm working on today concerning the ATP synthase. I thought that it would be important in this context to bring up a couple proteins that were not discussed in this context of confined water. One's aquaporin, and aquaporins, you find it across the tree of life. It's in animals, and mutations cause all sorts of nasty diseases. It's in plants. This particular one I'm showing is in plants, and it allows water to move across biological membranes in response to an osmotic gradient. And but it doesn't allow any of the ions to move across. So it's selective just for the water. And so on the left, you'll see that there's actually, it's a tetramer, so it has four pores that for each protein complex. Then in the middle, you're seeing some of the waters that are going down the middle in this water chain. And what the discriminating thing is shown up close on the right, where you have these residues that are coming together, and it's actually a very narrow pore, and that arginine is very important for causing the protein to work. And so there's a couple of different things of how it works. You have bodies of water on each side of the membrane that are coming down into the channel, but as it gets right to the middle of the channel, there's four waters that are shown in bold here. And this is where you get the selectivity, because the channel gets restricted down to 2.8 angstroms diameter, which is just about the size of one water molecule. So they have to pass one at a time through there. And then you have this electrostatic repulsion with the arginine 195 at that point of the narrow restriction. And it imposes a barrier to the cations, including, in fact, H3O, which is it's amazing to me that it can be that discriminatory. And then finally, the water dipole, as you go through from one side of the membrane to the other through this narrow pore, then as these two helices meet right there at that point by the arginine, you see that the dipole reorients of the water. So as it passes that point, and which I thought was very interesting, especially related to what Ali was presenting. So then Nathan talked about Marcus theory and how you have to have what are the criteria for that. And so in Marcus theory, then you will have it passing an electron from a donor to an acceptor, and it'll go downhill overall in energy. You'll see that you'll have two reactant surfaces, and then where they cross over, that energy difference is the reorganization energy. And so, and he quoted Schrodinger here at this point. I will say that last August at the International European Bioenergetics Conference that was in Austria, a group of us went to Schrodinger's grave. Okay, nerdy. And also we had dinner up by where he lived his life, up in the mountains, which is a beautiful place. Then Nathan talked about making these mutations, and Bethany pointed out what an elegant work this was. It really, really is elegant, and how, in fact, that the methionine protects those few waters that to essentially, again, constrain the number of waters that can be involved in order to enable the electron transfer to occur. And he compared this by making various mutants. He focused mostly on the water reorganization. There are two aspects actually of Marcus theory, and one is the driving force versus the rate, and that was tested by Les Dutton, and here there's a review with Christopher Moser, and but also Marilyn Gunner, who I mentioned before on the water oxidizing complex, was Les's grad student and also contributed a lot to this. And what they did was to vary the type of quinone that was in the photosynthetic bacterial reaction center. And the quinone had a different redox potential that they would substitute. And so that would change the overall energy and you could hit it with a photon and get the electron to jump over 20 to 25 angstroms across the membrane. And they looked at the rate of that as compared. And so when you do that, you're taking those two curves, those two surfaces, and moving one sideways to the other. And when you do that, then you're changing the point right there in the middle at which you have the reorganization energy to occur. And what they found, which was predicted by Rudy's models, was that you can get to, even though you put in a bigger, more driving force by changing the quinone, that the rate will go through a maximum and start to come down, a Goldilocks zone. And they called it the inverted region. And I often joked that I wished that my experiments had an inverted region, so that when the rate goes down, when I think it's supposed to go up, then it would have a reason for it. But so anyway, that the inverted region is shown on the right-hand side, where you can see these various quinones. And of course, then this is a really nice review article by Colin Raitt. And Colin looked at the actual waters in the quinone binding site that you can see right there in the middle, and to look at the reorganization energy. And my colleagues here in the Photosynthesis Research Center, which I'm a part, Jim Allen and company, they made mutations to each of those residues to change the energetics of reorganization. So to help and support Marcus theory that got Rudy the Nobel Prize. So yes, personal things. Rudy has, in recent years, been working on the ATP synthase and done theoretical work with that. Colin, in this review, he also looks at these constrained water channels in several proteins, and I showed the one in cytochrome oxidase. And again, to refer back to Ali, as well as several other people who were talking about constrained water and its effects. And I think this is Alistair talking about cytochrome oxidase. And I just showed one of those channels and greasing the turbines. And so one of the things that was discussed was, of course, ATP, but also temperature and having that interfacial water, the viscosity of it, so that things can move. And so, yes, these are important questions that are really being addressed today. I think this was Ali's talking about water dissociation near the mitochondria, its confinement, et cetera, and what's the role of pH on this functioning. And I'll talk about that in a minute as relevant to my own work. But yes, these are important things that are being discussed right now. I thought Nathan was courageous to talk about, sort of refer to my work. He didn't really talk about it too much, but he talked about this Morelli paper. And I would say that this figure shown in Morelli's paper, I just have to disagree with it. Early in the, I would say, in the mid 1970s, then Dick Dilley had this idea originally, and it was a lot of arguing over it. A lot of experiments were done, and it was pretty much shown not to be the case. One of the things I did go through the Morelli paper, and he does refer to some really recent work. At the time that Dick Dilley was talking about this, it was like, oh, how come the protons can move so fast across the membrane? And so there has to be this connection somehow. And so at the time, people did not believe any of the proteins in a mitochondria were connected to each other. But now and Morelli points us out that there are these super complexes of complex 1, 3, and 4, so that the electron transfer can occur very rapidly and the protons can get pumped. But no, I don't believe they go through the middle of the membrane. That's been refuted, and there's been no evidence at all to date that the ATP synthase and complex 1 make a supercomplex, and people have looked quite a lot at this. But never say never. This could happen. I will, though, spend some time, if you go to the next slide, talking about my own work in this regard. And so the ATP synthase, I'm showing it here a lot of times. I show it upside down compared to how a lot of people do, because when I mount it onto a slide to do single molecule work, then it essentially is in that configuration. And I attach a visible probe so I can watch it rotate. And so the protons come in from what I'm showing here as the top into an input channel, and the input channel is in subunit A, and they're transferred over to the C ring. There's a carboxyl group on each C subunit, and then they have to rotate all the way around in the membrane, and then they're delivered back to subunit A, where there is an exit channel. And over in the far right, I've shown a cross section looking down on the subunit A, which you can see is over on the left of that, and the C ring is on the right, and you can see the carboxyl groups as they come in. The residues that were involved were identified quite early on: the D119, E219, H245, N214, and Q252. It was later on the output, that was from the input channel. In the output channel, then the S199, and there's also an E196 down at the bottom. Those were established recently. E196 was work that I did to do that. So these are some of the single molecule studies that we did. We drove rotation by ATP hydrolysis, so the energy was more than what ATP synthesis could do. But even so, we found that instead of just rotating continuously, we saw these breaks that you can see on the far left by these little red lines. And in fact, the FO portion could drive things backwards from the ATP hydrolysis-driven rotation to go in the ATP synthesis direction. And based on this, we did this as a function of pH. We established that the input channel has a pKa of 5.6, while the output channel has a pKa of 7.5. So even without a proton gradient, you still are populating the input channel with protons more than you are the output channel. So you're getting a delta pH across the membrane just by the difference in the pHs. And then if you go over to frame, let's just jump all the way to frame E, and where you can see that the extent to which it rotated in the ATP synthase direction, those red lines on the far left, that always came out to be 11 degrees. Now, we made mutations to all of the residues I told you that were known to be participating in the proton transfer pathway through subunit A, and they changed the pKa values. And no matter whether it was on the input channel or the output channel, it changed both of them. So we concluded from that the only way that could happen was that they acted as one channel completely that was tied together by the rotation of the C subunit. And so the blue in F, if you look at the top, the blue line shows the amount of protonation of the pKa1, and the red line shows that of pKa2. So or unprotonated. So that's the output channel where it's unprotonated, and the blue one is input channel. And it's where those cross at that pH where we saw the largest amount of this ATP synthase going. So based on this 11-degree step and the fact that they were working together, we concluded that the proton flow must be occurring by a Groothuis mechanism. You can mutate these residues and you still get the function, but it just changes the pH. So those residues aren't actually necessarily causing the movement of the protons directly. It must be a water channel in there. And so we have a proton-dependent rotation, but it's only 11 degrees. The E. coli ATP synthase has a C10 ring, so that the movement of each C subunit would be 36 degrees, but the proton-dependent one's only 11. But what that does is moves the carboxyl group out from the membrane, which is shown as the yellow, and it moves it into this, what we propose to be an aqueous vestibule, where it's now stabilized by the negative charge of the unprotonated group is now stabilized, and that gets attracted to the positive arginine, and the electrostatic attraction then pulls it the remaining 25 degrees, and then it repeats. And so, so this is what we're continuing to work on, is this interplay between these fixed waters and these mobile waters in these vestibules. And of course, the amount of water in there is going to change in the vestibule, is going to change the dielectric constant, which will pull the negative charge to the positive charge. But if it's too hydrophobic, it'll pull it together, but you won't be able to get them apart. So again, it's one of these Goldilocks zone kind of things that we're working on to understand how it works. In just the last few months, that Werner Kulbrandt at the Max Planck in Frankfurt has gotten a structure that actually resolves the waters in the Groothuis channel that I had predicted. And so he and I are starting to collaborate on this. You might also see that I have this periplasma funnel. And what is that about? So Colin Wright, he was looking at the rate of proton transfer through the FO, and he said it can go way too fast for the ability of the protons to make it from complex 1, something Morelli was talking about. So how would this happen? How do you pull this out? And he said, well, there has to be some sort of an antenna there, a proton antenna that sucks them in, and to pull them away and desolvate them out of the bulk water. And so he actually, based on the rate of proton transfer through FO, he predicted what the size of that antenna was. So, and he did this before there was a structure. I looked at the structure and I able to find this. If you look, the A is on the side view and the B is looking down, and we actually do see this funnel, and the funnel, even though it's down in the membrane, the funnel protrudes up to the aqueous phase, and it's decorated with charged and polar residues that can help move those protons at low concentration out of the aqueous phase, the bulk phase, and into the channel. So the Groothuis mechanism. There's a really nice paper about Groothuis by Samuel Kuckerman, and he actually found the original paper down at the bottom by Groothuis, which was published first in 1806. And he didn't actually propose it as a means of moving protons quickly, but it has come to be that way. So here at the top, over on the left, you see a series of waters that are all in a confined space, and they are hydrogen bonded and linked together in this manner. And then you add a proton in, as you would if you're entering into that from the input channel of our FO, or some of these other channels that I talked about earlier, described by Colin. And so what happens then is that as one proton comes in, they're all connected, and all you have to do is change the hydrogen bonding to move that proton. So one comes in and immediately a proton comes out the other end. But what that does is that it rearranges the orientation. So you have to have the waters all convert back to their original orientation to go. So there has to be some coherence among these so they all work together. And then it's been postulated, is this turn that they show, is that reorganization energy? It's not determined yet. The poster child for the first Groothuis channel was in Grammacidin. You can see the waters in the middle of that going through that channel. Again, it's confined water and how it behaves. And so I think I'm getting to the end here. And so both Bob and Alistair talked about this spectral irradiance and how where photosynthesis occurs in the visible light, but how an awful lot of infrared light is very important and to this process, and that of course in Alistair had a nice little thing of the waters moving back and forth in their vibration rotation modes. And so it's very possible that the infrared light is helping to move the protons in the mitochondrial membrane along these surfaces of the surface of the membrane of water that doesn't really want to move very much, to help move those protons from complex 1 down into the FO complex. And it occurs to me that perhaps that there are specific infrared wavelengths that might facilitate this movement more than others, based on, if you look on the right there. So I think this is it. And hopefully I've brought up a number of topics that people want to discuss.